talking about some work that we've been doing. So I'm in evolutionary biology in the Department of Animal and Plant Sciences. I've been in Sheffield for the last 3 years, setting up my own research group. Before that, I was a postdoc in UCL in London, and then before that, I completed my PhD at the University of Oxford. So really broadly, my research interest is centred around trying to understand how and why sex differences across the animal kingdom evolve. And so when we look across the tree of life, males and females of the same species can be strikingly different. So not only in terms of really basic reproductive traits, but a whole host of other characteristics. And these sex differences across the animal kingdom are really common, and therefore they make up a really important component of biodiversity. And so I'm really broadly interested in trying to understand how and why that diversity arises. So sexual dimorphism can actually be really, sometimes really profound. And this is my favourite example of sexual dimorphism. So do any of you guys know what this is? What did you say, sorry? Yes, exactly. So this is an anglerfish. So this is a female. So it's essentially a deep-sea marine fish that lives at the bottom of deep oceans where it's really dark, and their individuals are very widely distributed across the ocean floor. And so because of that, it means that individuals very rarely interact with each other, and obviously that has really important consequences for mate choice and for finding a mate if you very rarely interact with a member of the opposite sex. So to kind of counteract that, females have evolved this bioluminescent organ, which essentially emits light that's useful for attracting not only prey, but also is useful for attracting mates. So this little guy here is the male anglerfish. And this is why I love this as an example of sex differences, because they're really extreme. So not only is there obviously these huge differences in size, yes?
How big are we looking on the real scale?
Yes. So the females are about this big, and they're tiny. Yeah, teeny tiny. Yeah. So you get this huge difference in body size, but they also have really different behaviours. So essentially, when a male encounters a female, because as I said, individuals don't often interact frequently with each other because of the environment they live in, the male really doesn't want to miss out on this mating opportunity. And so what they do is the male swims up to the female, burrows into the female body cavity. All of his organs completely dissolve, leaving only his reproductive organs intact, buried, as you can kind of see here, inside the female's body cavity. And essentially those gonads, which are just stuck inside the female, release sperm, fertilising the female's eggs, and she just swims around, essentially with all of these males embedded in her body. And for ages, it was actually thought that these were two separate species, because when fishermen were kind of pulling up these fish from the deep, they thought that this was the anglerfish and that these other organisms that they saw were actually just a parasitic species that was just parasitizing the female. And it was only much more recently that we realised that males and females, that they're actually just males and females of the same species. So clearly, sex differences, really important component of biodiversity across the tree of life. However, sex differences don't always necessarily have to be quite so profound. Males and females can differ in a whole host of other characteristics, including how they look, how they behave, their physiology, the types of diseases that they get, how you age, how males and females age. And so in this example, this is the sailfin molly. This is the male, and he has this really elongated dorsal fin, and females really like to mate with males with these elongated fins. So the bigger the fin, the more sexy the male, essentially, for the female. Similarly here, this is a bird called a ruff, which is a territorial wading bird. And males have these really fantastic, elaborate set of plumage feathers around their neck, hence why they're called a ruff. And they use these in the breeding season to display to females to attract them to their territory. So as you can see, males and females and this sexual dimorphism can be very common and differ in a number of different ways. So I'm really interested in understanding how and why these differences arise. And so specifically, I'm first interested in understanding the mechanism by which sex is determined, so that trigger that determines whether an individual develops into a male or a female. But then also, once sex is determined and established, there's the potential for some individuals to be more attractive or more beautiful to the other sex than other individuals. And so I'm interested in trying to understand why that variation arises. So lots of the research that I've done so far focuses on fish, and that's what I'm going to be talking about with you today. But we're not just limited to fish in my group, so we've also done some research in birds, and we're also trying to develop some insect species as a model for looking at some of these questions. So sex and sex determination mechanisms have evolved countless independent times across the animal kingdom. And so recently there was this Tree of Sex consortium that essentially set out to kind of condense all of the information we know about how sex is determined across the tree of life. So they were then able to map all of that information across all of the species that we know, where every single colour is essentially a different way in which, or a different way of determining whether an individual develops into a male or a female. And so you can really see that these mechanisms are really diverse, and it's actually likely that we're really underestimating this diversity, because in many species we still don't know how sex is determined, so we still don't understand what that trigger is that determines whether individuals develop into a male or a female. And so you're probably most familiar with genetic sex determination as a mechanism, where essentially there are genes on sex chromosomes that determine what sex an individual will develop into. So that would be XY sex chromosomes, so ZW sex chromosomes, which I don't know if some of you have heard of, where it's the reverse to XY, so males are ZZ, females are ZW, but it's again the presence of a gene that determines whether you develop into a male or a female, and kind of genetic sex determination in yellow.
In the plant kingdom, is there an equally diverse array?
Yes, it's yes, they don't have some of these weird mechanisms. So, for example, like in these scale bugs here, they have this thing called paternal genome elimination, where essentially all of the genes that you inherit from your father just are ejected from the cell, and then that determines essentially the sex. So they don't have some mechanisms like that, but they have XY, ZW sex chromosomes, and it does seem to also evolve, quite quickly. That's one of the things.
I was just interested because I was wondering if these are coming from behavioural mechanisms, and then what the plants have behavioural mechanisms.
Yes. Well, I mean, I guess there is, like environmental triggers as well, but So actually, one of the things I was going to talk about next was, the presence of essentially environmental triggers to determine sex, and so that's everything that's in green, so it's really common in reptiles, in fish, and again, some of these scale bugs. And this is where essentially you have some environmental trigger that determines which sex you develop into. And so there'll be some regulatory molecule that responds to environmental conditions that will then trigger transcription of the gene that determines sex, leading to kind of the cascade of the sex determination pathway. So for example, in American alligators, it's the temperature at which the eggs are incubated that's important. So at around 33 degrees, the clutch develops into mainly males. At about 30 degrees, it's mainly females. But I really like this example of the blue-banded goby, where it's essentially social behaviour and social dominance that determines sex. So they have, in this system, a dominant male that is very aggressive, and that aggressive behaviour essentially suppresses females from developing into males. So when that dominant male is removed from the population, the largest female immediately changes behaviour and starts to produce sperm in about 3 days. So again, the mechanisms even within lots of these are extremely, very variable. So one of the things that I'm sure that you've noticed about this is that not only are there a large number of different ways in which sex can be determined, but they actually seem to be quite variable across different groups. So, for example, in mammals and birds, they all have the same sex chromosomes, so it evolved in the common ancestor of those groups, and every single mammal has the same XY sex chromosome. But you can see in reptiles and fish and in crustaceans, there's really, really variable mechanisms. So very closely related species can have independently involved mechanisms. So one could be XY sex chromosomes, and then a very closely related species could have an environmental trigger. And essentially, we don't know why this is. But clearly, it's really important because it seems to vary a lot across these different groups, but we don't have an answer for why this variation exists. And this is kind of like a hot topic in evolutionary biology at the moment, and there are lots of different groups kind of trying to answer this question. So I'm really sorry I can't answer this for you now, but hopefully at some point in the future we will be able to shed a bit more light on this. So you're probably most familiar with the XY sex chromosomes of mammals. So all mammals have the same XY sex chromosome. And so if you imagine all of the cells, all of the chromosomes within a cell, you can see that the sex chromosomes look very different from the other chromosomes. So the Y chromosome here is really tiny and it's really degenerate compared to the X chromosome. And so this Y chromosome contains a gene called the sex-determining region on the Y, and that's essentially the trigger for maleness. So they look really different, but actually we think at one point that these chromosomes were identical, and that they contained exactly the same number of genes, but that over millions of years the chromosomes have diverged and become different, and the Y chromosome has degenerated and lost lots and lots of genes. So you might have seen some of this in the newspaper a couple of years ago, but this led to quite a number of sensationalist headlines suggesting that the human Y chromosome initially had loads of genes, so it had about 700 genes initially, but over millions of years those genes have started to degenerate, it's lost genes, it's become a bit decrepit, there have been huge deletions on the Y. And so the logic that kind of lots of these newspaper articles was following was that if this process of gene loss and gene degeneration continues, so at the moment in humans there are only 45 genes left on the Y, whereas originally there were 700. So if this process of loss continues, then eventually the Y chromosome will completely disappear. If the Y chromosome completely disappears, then males will go extinct, and then what will happen to the human species? But luckily, most evolutionary biologists think that this probably isn't the case, because the Y chromosome, at least in humans, has a very important role in fertility and reproduction. So what's more likely is that there's a very initial rate of gene loss, and then it's much more gradual as these genes that remain are really important for, say, sperm production, and so they're preserved on the Y chromosome because they're just really important, and so it's kind of maintained these remaining genes. Yes?
What were the genes that have already been lost? What were they coded for?
Yeah, that's such a good question. So that's something that we're starting to look at. So it's difficult to say what a specific gene does. So there are lots of genes across the genome and we don't know all of their functions, but they didn't do anything really different from what genes on other chromosomes do. So they probably played just roles generally in kind of cell cycles and just no function, nothing that kind of really sets them apart from any other chromosome. But I think yours is such a good question because it kind of raises the idea of maybe there are some genes where it's more likely that they're lost than other genes. And so that's what we're starting to look at, why are the genes that were lost? Is it because they were less important, or is it just random? And you just have a random chance of being lost. So yes. But all we know at the moment, because this happened millions of years ago, so it's really difficult to look back and say what did those genes do, which ones were lost. So all we know at the moment in human Y is that there's about 45 of these genes remaining. They're probably really important. Well, we know some of them are really important in sperm production and fertility, so they're probably not going anywhere, which is good because it obviously means that males are unlikely to go extinct.
I'm going to really expose my biology knowledge now, because I always assumed that the genes on the X were pretty much the same as the genes on the Y, because they're almost like a homologous pair.
Yeah, well, they were originally, yeah,
right
at the beginning.
But now they're so that they're quite so the genes for sperm production aren't just silenced on the X chromosome.
No,
they're just not there.
no, they've like evolved new functions essentially on the Y.
So sorry, this is going to completely go on a tangent now. But so when in meiosis, which you guys are all learning about, when they all pair up as homologous pairs, what sticks the X and the Y together?
Yes, so they have a really tiny region called the PAR, which is called the pseudo-autosomal region, which is the bit that is still the same between the X and the Y.
Right.
And you need that in order to be able to pair.
Awesome.
Yes. But in some species the PAR is really teeny tiny, in others it's massive. So
I've gone through my entire career of biology completely unaware of all of what you've just said.
Well,
so that's
you're
brilliant.
right for some species, just not humans.
Oh, okay.
Some of them it is similar.
Thank you. So I haven't been wrong. Brilliant.
Yes?
So would the same genes on the Y chromosome be on the X chromosome as well? So could we change the X chromosome to replicate the Y?
So some genes that are on the Y could be similar to the X chromosome, but because they've been separate for such a long period of time, like millions of years, they're kind of evolving to do their own thing. So the idea is that the Y evolves to be important for male function because it's only ever present in males. And actually there's a lot of theory suggesting that the X should be really important for female fitness because it's more often in females than it is in males. So again, it kind of varies, but some genes will be more similar, but other genes are kind of just completely evolved their own functions.
Brilliant.
Yes, so following on from this, we were kind of interested in more explicitly testing the role of the Y chromosome in maleness, and also, again, this idea that it's going to go extinct. We were interested in looking in other species. And so we were doing that in guppies. So guppies are kind of a tropical fish. You've probably seen them in pet shops because they're really common there. This is the female, and these are males. So the female is much bigger than the males, and she's really dull in colour, whereas males are super colourful. And females really like to mate with colourful males, so they find colourful males really beautiful. And there's two particular things that they like about males. They really like the colour orange for some reason. So people have looked at preference studies of guppies. You give them a choice of different males, and you say which one. You essentially see which one they prefer to mate with. They like males with orange colours, and they also like colour patterns that they've never seen before. So if you give them the option to mate with a male with a colour pattern that they've not seen before, they will prefer that. So they like orange and they like novelty. Again, not really clear why that is. So they also have XY chromosomes like humans, not the same XY pair, but it's the same mechanism. And so this is essentially all of the chromosomes in a guppy cell. And the first thing that you can see that looks really different from the cell that I showed you from humans before, so these are the sex chromosomes here, is that the Y chromosome is massive compared to the X chromosome. So it's really huge. It's like almost twice the size. And so this is kind of the starting to answer our question of whether Y chromosomes are going to always go extinct. Well, clearly not in this case because it's way bigger than the X chromosome. And so we were then interested in looking, okay, what is this Y chromosome actually doing? Is it important for male attractiveness and sex differences? So guppies are a really nice system for starting to test this because in the wild there are different populations of guppies that vary in the amount of male coloration and the amount of female preference for colour. So in northern Trinidad you have different watersheds, and in each watershed there are upstream regions and there are downstream regions, and they're separated by waterfalls. And guppies from the downstream regions have managed to get up these waterfalls into the upstream regions, and there are upstream populations of guppies and downstream populations of guppies. So in the upstream populations, because it's difficult to get up these waterfalls from downstream to upstream, not many other predators or fish have done that. So there are fewer guppy predators in the upstream populations than there are in the downstream populations. So because there are fewer predators, it means that males have evolved to be really colourful, And females really like to mate with these orange males that have these novel colour patterns. Whereas in the downstream populations, there are loads of predators, and therefore, obviously being colourful means that you're going to just be eaten immediately by a predator. So males haven't evolved to be colourful, and females also don't like to mate with colourful males, because if they do, their sons are going to be colourful, and then those sons are going to be immediately eaten by predators. So what you get is in the downstream populations, you get these much less colourful males, which I've kind of denoted with this grey colour, whereas in the upstream populations, males are really colourful, and females like to mate with these really colourful males. So essentially, we had some collaborators. Unfortunately, I was not able to go, even though it would have been amazing, who went to northern Trinidad and essentially sampled males from each of these 3 watersheds, from upstream populations and downstream populations. And the first thing that we did was we essentially built a family tree for these 6 different populations of guppies. So the first thing that you can, and so essentially this shows relatedness across those different individuals. So what we see is that guppies are more closely related to each other within each watershed. So that's Yarra watershed, Aripo watershed, and Kwara watershed. And the upstream individuals, or the population, has evolved from the downstream individuals. So there's essentially been downstream guppies, they've managed to get up the waterfalls to the upstream populations and then become more colourful. So what we then did was looked at the sex chromosomes of the guppies in these different populations. So this is in the dull downstream populations. This is essentially when we sequence the sex chromosomes, kind of a schematic of what it looked like. So you have the sex-determining region, which is essentially Y-linked. So this is the Y bit here, kind of at the tip of the chromosome. And then this is the pseudo-autosomal region, which was I was just talking about, which is essentially identical between the X and the Y. It recombines, it pairs, it doesn't have anything to do with sexual dimorphism or sex determination. So this is the kind of Y-linked region. So we were interested in asking, well, if Y chromosomes are important for male colour and sexual dimorphism, is this region bigger in the colourful populations? And it is, which was amazing. We were so pleased when we saw this. So it's quite a bit bigger in each of those colourful populations. So what this means is that this sex-determining region has essentially expanded independently 3 separate times across these colourful populations. And we think that this is because in this region here, it's expanded to essentially contain all of those genes that are really important for male colour. And so that's why you see the colourful populations upstream having this much bigger region, because it's essentially how this colour is encoded, because it's on the Y chromosome. And so this, we were very excited about this because it seems to be quite a important force in selection, because it happens this 3 independent times, and we can see consistent and striking differences between these different populations that really they only differ in the amount of male colour.
Are they the same species?
Yes, just different populations. So we think that there is some gene flow between the populations, but it must be so strong, there must be really strong selection against it to kind of maintain these really consistent differences between the different populations. So, also linked to that question, we were interested in looking at other species and saying, well, what's going on across guppies more generally? Because there are lots of different guppy species. So this is the species that I just showed you the results for. There's a very closely related species called wing-eye, which is really colourful. Then there's Picta here, which is much less colourful, so that evolved about 30 million years ago. Sailfin molly, and this is the mosquito fish. So again, we took samples from males and females of each of these different species. We identified the sex chromosomes, and then looked to see how they vary across these different species. And again, we were super excited because it's really, really diverse, and they've evolved new sex chromosomes in this group. So these 2 guppy species have XY chromosomes, and it's the same XY. But then you move to only about 30 million years ago. Picta lost its Y chromosome, doesn't have one. Not entirely sure why, but it has these really diverged sex chromosomes where males just have a single X. Then you move a little bit further back in evolutionary time to the sailfin molly. It doesn't have XY sex chromosomes, it has ZW sex chromosomes that have evolved from a completely another pair of autosomes. And then you move here to the mosquito fish, which has XY, but it's not the same XY as the guppies up here have. So again, there's huge turnover and diversity in the evolution of sex determination and sex chromosomes across this system. And you compare this to mammals, which are much older than this. We all have the same XY chromosomes, all different species. So why this diversity exists here, but not in other groups? Again, this is something that we're looking into. So essentially, this classic idea of you evolve sex chromosomes, and over time they gradually diverge, and then the Y chromosome gradually decays, and then it's eventually completely lost. This is kind of the classic view of sex chromosome evolution. It turns out that that's not really the case anymore. And it's a much more dynamic and fluid system that we can kind of think of like a cycle of sex chromosome evolution. So many species will go through the stages of the evolution of sex chromosomes, divergence, more divergence, start to degenerate and lost. That does happen in some species. However, at each stage, those sex chromosomes can persist. So the pufferfish, it has one gene that's involved in sex determination on the sex chromosomes. Everything else is identical, and that can persist for millions of years. You can go back and forth between these different stages, so it looks like you can actually undo some of the degeneration that we observe across some species. But at any time, it looks like you can essentially completely lose your sex chromosomes and evolve a completely new sex chromosome pair, like we see across the guppies. And so we're starting to get kind of a grip on why you get this in some species, but it differs in other species. So I kind of spent that section talking about sex chromosomes, their evolution, why we think that they might be really important for sexual dimorphism, and kind of the evolution of beauty or attractiveness. However, you can see from this cell, the sex chromosomes are really small. There are loads of other chromosomes. And actually in many species, such as those with environmental sex determination, they don't have sex chromosomes. And so this then poses a really fundamental problem for evolutionary biologists, which is if males and females have exactly the same DNA and genome, how do sex differences ever actually arise if they have exactly the same genetic information? And so that was something that we were interested in starting to look at. And so we did that using the ocellated wrasse, which is a kind of Mediterranean fish. So this is a really nice system for starting to look at that question because sexual dimorphism is quite complicated in this system. So unlike in many species where differences between male and female phenotypes are quite discrete, and there's male phenotypes and there's female phenotypes, in many species it's much more complex and there's actually more of a gradient of sex differences. And you can see that really nicely in the ocellated wrasse. So this is the female here, and these are all males. And they're all males, they all produce sperm. So this is a male called a nesting male. So he's really big compared to the female. Is really colourful. Nesting males build nests. They court females. Females swim into the nest, they lay their eggs, and the nesting male looks after those eggs until they essentially hatch. But you also have a satellite male, and the satellite male obviously looks much less kind of colourful, more like a female than the nesting male. And this satellite male essentially hangs around with the nesting male, and sometimes when the female swims into the nest, the satellite male will also swim in and try and fertilise some of the eggs that she's depositing. But the nesting male is okay with that because females really like to mate with nesting males, but they really like to mate with nesting males that have loads of satellite males. It's almost like they have wingmen to essentially say, I'm a really good quality male. And then you have another type of male, which is essentially called a sneaker male. And this sneaker male is essentially identical in appearance to the female. And what the sneaker male does is he tries to trick the nesting male and the satellite male into thinking that it's a female, and then can essentially swim into the nest and sneak mating attempts. And so you can see here it's not a discrete continuum between male and female phenotypes, but a discrete difference between male and female phenotypes, but there's kind of this continuum of sex differences. And this is really nice for starting to look at actually the genes that are important for understanding sex differences. So these guys don't have sex chromosomes, so all of these different, the females in these different morphs, they all have the same genetic information. Yes?
Do the different morphs of male, can a seeker become a nester?
Yes, they can.
Oh, okay. Yeah.
Yes. So, yes. It's all to do with how old they are and also to do with their quality. So I can't remember the exact order, but I think they all start out maybe satellite, and then in the second year you can change to a nesting male depending on your quality. I haven't worked like explicitly with these groups, these morphs, but we have a collaborator who collected the samples for us, and she's essentially interested in trying to understand like when you switch from one to the other. But it's complicated.
Yeah.
Yes.
You say quality. What do you mean?
Pardon?
What do you mean by quality?
Oh, like how healthy you are. Sorry. Yes. Yeah. So like if which is sometimes like a size threshold. So it might be like that you can switch from satellite to nesting when you're 2 years old, but you only do it if you're really healthy.
Okay.
Yeah, which might be indicated by like body size. So if you're healthy, then you've grown to like a certain size, and then there's a threshold that you then switch. Yeah. So they all have the same genetic information, so we were then interested in looking at how differences in how the genes are used between the different morphs might be important. And so we measured differences in gene activity or gene expression across these different individuals. So then we essentially were plotting gene activity for all the genes that were shared. Well, yeah, they have essentially the same genetic information. So and then we were plotting it as a heat map. So every single column is a different individual. So these are the females, these are the nesting males, sneaker males, and satellite males. And every single row is a different individual. Every single row is a different gene. And if the gene is really active, so it's expressed really highly, producing lots of product, it's in yellow. But if it's really lowly expressed or turned off, it's essentially in black. And you can see that there are quite striking differences between the different morphs, mainly between the females and these different males. But then you can see that the males don't just randomly cluster across these different types. Like all the satellite males cluster together because they have similar expression levels. Similarly, all the sneaker males cluster together because they have similar expression levels. And so we think that this is indicating that actually differences in the expression of genes that all of the different individuals share is actually what's really important in establishing these differences in phenotype. So it's essentially, in this case, it's not the evolution of sexual dimorphism and the difference between male morphs is not so much to do with the genes that you do or don't have, it's to do with the way in which those genes are used differently across different individuals. So in that case, gene expression is really important for establishing those differences.
So is that a form of epigenetics then?
Yes, yeah, exactly, yes.
That's what you'll be looking at next year. It's into the A-level spec now.
Oh, good. Oh, that's great.
Mm-hmm.
Awesome. Yeah, so actually, because this is one thing, like when you measure gene expression, it's quite a crude way of looking at these differences, and there are actually lots of different types of expression. So I think, again, digging down and trying to understand what's actually causing this would be very cool. So again, you can see that the males do cluster into these three different groups, which are consistent with kind of a nesting male group, satellite male, and a seeker male group. So yeah, that's it.
Super.
I hope you've enjoyed it.
There was lots of questions in there. Does anyone get any other questions?
Yeah.
That was really fascinating. Thank you. I love that.
It was good, great questions.
Anyone got any other questions? Richard?
Yeah.
What's the primary difference between why they're females and why they're males? And why do we have two different sexes?
Yes, that is a great question. So some of it is to do with the evolution of kind of gamete strategy. So individuals can essentially produce a gamete, but at some point it's advantageous to produce really small gametes, because if you can produce, when I say gamete, do you guys, yeah, okay, awesome. At some point it's really useful to produce a really small gamete, because if they're small, you don't have to invest in as much of them, and then you can produce loads. But at the same time, there becomes a trade-off between how small they are and actually the advantage of producing a really big gamete, because if you produce a big gamete, then essentially it has greater chance of being fertilized by the really small gametes. So there's like disruptive selection for some individuals to produce really small gametes where they can produce loads of them, and some individuals within a population to produce one or like a couple of really big gametes. So that then establishes two different like reproductive strategies. And if you produce a really big gamete, that produces like a lot of energy. That generates a lot of energy, and you should probably be really choosy about who you mate with, because all of your kind of reproductive investment is being put into like a couple of really big gametes. So that's kind of like the female strategy, and then there'll be selection acting on those individuals in order to kind of maximize other things that are associated with that. So being choosy about who you mate with, putting lots of energy into looking after that egg and making sure essentially that it's that the investment that you've put in is not a waste. So that would then be associated with like parental care. Whereas in the other strategy, which would be producing lots of small gametes, would be typically associated with a male phenotype, which would then be multiple matings to try and maximize the number of individuals that they can fertilize. And so that's initially why we think that you get two different reproductive strategies, but there's loads of variation in relation to that that's laid on by ecology and differences in the environment. So again, like whether you can monopolize an environment as a male will determine how choosy you're going to be and how much investment you're going to put into kind of parental care. But yeah, that's initially why we think those differences. It's essentially like how you invest energy.
So in terms of those two strategies you just described, is it like fish that produce loads?
Yeah.
Whereas your mammals and your birds have a large gamete, but in mammals and birds there's not that many different sex determinations. So
yes.
is that one of the reasons why because of those two strategies, you only get all the variation when you have this mass approach?
Well, so I guess it's to do with males and females within one species. So like birds would be a really extreme example where females produce like a massive egg and males produce lots of... Whereas I guess it's kind of similar in fish, where females do still produce much bigger eggs. So, because with your first graphic, you've got the kind of the reptiles and the fish and the insects, which probably have the mass strategy, and could be completely wrong, but I just sort of can see that link. I guess you need to look in some species where they don't have those huge differences in gamete size. Like there are some algae that don't, but they still have a lot of turnover in sex determination mechanism. But
it's also like the parenting thing as well, isn't it?
Yes, so there is an idea that, because parenting is, like parental care is meant to be linked to sexual selection. So again, it's to do with the amount of resources that are available to you and whether it's more beneficial to invest in paternal care, and if it's more advantageous to mate multiply all the time, then that might be indicative of higher levels of sexual selection, and then sexual selection might be important in driving like turnover of sex chromosome systems. So yeah, they're all kind of interlinked. But the thing that's nice about it is there's so much variation. Like that's just kind of some examples of how it works in some species, but it's just completely different in others. Yes.
You know, boom primates to start with the anglefish.
Yes.
So the male's never going to die.
Yeah, I guess not. I mean, I guess it's their most the strategy that evolution has favoured over millions of years. So actually it's best for their fitness to do that.
Okay.
Because if you think about it, if they didn't do that, say they met a female, they would mate with that female, then they might not see another female for X number of times.
Yeah.
Whereas if they are essentially like just a gonad stuck inside the body, then they're essentially producing loads and loads of offspring. And I guess that's the most important thing for natural selection to like act upon, the number of offspring that you produce. So males that do that strategy will produce way more offspring than males that don't do that.
Yeah, okay.
There's no opportunity for them to say, "Oh, this female's got lots of males embedded in her. I should probably choose a different female."
It's a good point.
Because of the
That
competition.
would be very cool to look at. I think they're so difficult to study because of their weird ecology, but you'd think that there would be something where if a male can find a female that doesn't have loads of males, then there's less sperm competition, and so therefore they might not.
But the whole strategy relies on the fact that there isn't very many females, so I suppose you wouldn't hang around, would you?
Yeah. Yeah, that's true. But again, I guess it's what would be like the most fit strategy.
That's really fascinating.
Yeah. Oh, no.
Bit of biology that I think I might have been pretty much unaware of. So thank you.
So, yeah.
Any more questions, folks?
Yes.
Oh.
Could you discover any mechanisms for when the determining sex changes from XY to ZW?
Yes, so there are examples of that. And so I'm trying to think. So there are geckos, they're really weird. They're always switching between XY and ZW, different species. In some gecko populations, or in some gecko species, you actually have some populations that are XY and like other populations, so it's the same species that is ZW. But again, unless you capture that exact turnover event when it's happening, which is like very difficult to do because it happens over millions of years, it's really hard to know why it happens, because you're essentially like extrapolating back in time, as with all evolutionary studies. But yes, it definitely does.
Can you do that patent allowed by
Yes, that would be the coolest thing. If you can think of a way to do that, then I would love to hear it. But yeah, that would be the ideal. I don't know how you do it.
Can I ask one more question?
Yeah.
Sorry. So you've had the fugu up. Now, I was in a lecture many years ago, and they were sequencing the fugu because it doesn't have much junk DNA.
Okay, cool.
So you've said there that you pointed that out as there being there's only one gene.
Yes.
So that's then got me thinking, do different organisms, different species have different levels of sort of mobile genetic elements which can shuffle the DNA?
Yes.
And therefore the shuffling of the DNA leads to more turnover and therefore more loss of sex chromosomes.
Yes.
Do people look at that?
Yes. Yes. So no one has explicitly tested that, again, because it's difficult to link it directly with, it's difficult to extrapolate back and say this was the number of mobile genetic elements when this turnover event happened. But it's a good point. I'm not sure if anyone has tested that. Yeah, I mean, it would tie it, so birds don't have active transposable elements. Their genome is really static, and that would tie in with them having really stable sex chromosomes. But mammals have lots of things jumping around, and they also have stable. But yeah, it would be cool to look at that. Yeah, that's a good idea. Yeah.
Awesome. Okay. Brilliant.
Awesome.
Thank you. Go
yes.
on. More.
What happens with like plants, where you've got parts of plants male and parts of plants human, the same organs?
Yeah, so you can, so I want to...
Like when it's growing, how does it specialise in sort of
so you mean when you have a plant that has like male and female flowers? Yes. So they just do how a cell's fate is determined differently. So those species, some of those will have, so plants are weird in that you can have species where males and females are separate plants. Then you can have species where there are separate male and female flowers. And then you have some plants where there'll be hermaphrodites and they have both male and female reproductive organs within the same flower. So they don't have sex chromosomes. They're like, they essentially just produce both males and females. So there's just some trigger. Well, so essentially, but all cells will be the same, so they just produce, both males and females. I mean, it's the same with like nematode worms, they're hermaphrodites, so they produce worms that have both male and female gonads. So it's just another way of kind of doing sex. Instead of partitioning it into individuals, you essentially just have it all within the same individual, which can be beneficial because it means that you essentially always ensure that you're going to produce offspring because you just self-fertilize. Yeah, but actually just related to your thing, you get, this is not something that is like a, but so it's like a cell division error, but in birds, you can get something that goes very wrong early in development where so if they're, say, ZW, in some of the cells, they lose, say, the W chromosome. How does it work? But essentially, you end up with, like, there are pictures of chickens, which straight down the middle, this is male, this is female. I really want to see if I can find, like, I'm not sure my phone has, if you, I can find a picture of it, it's really easy. It's called a gonadromorph, and it's just something that happens very early in development, and something goes wrong, and this is genetically female, and this is genetically. So
it's like literally in a zygote, this half the embryo becomes that's bonkers.
So just with the ZW, or can that happen with XY?
So it's not to do with ZW, it's to do with the way that sex is determined. In birds, it's much more on a cell-by-cell basis, whereas so they don't have hormones in the same way that mammals do. So in mammals there'll be a hormonal cue that will influence those organs or whatever to develop into like a male or a female, whereas in birds it's more like cell by cell, so it matters the chromosomes that you have in the cell. Yes.
Self-replicating organisms, what determines their sex?
Well, they don't have sex because they're clonal, so they're asexual. So they just essentially divide and then so I guess in that sense you could say that they were female, I guess. But there's not like two sexes, there's just one which just clones itself. Does that answer your question? Yes, exactly. So there are some reptiles that do that. So that's kind of, I think they're probably female, but it's asexual, so there's no sex involved. They just copy themselves.
Right, we've got to stop asking questions. I don't want to stop asking questions because this is the most questions and discussion we've ever had. This is brilliant, but you guys have got to get home. So thank you very much.
No,
brilliant.
It was great. And if anyone else wants to